Thanks to you and the sponsors for putting on this program. I think it's wonderful to be right here in the state of Wisconsin. And I'm really honored to be here with all the other great speakers, myself not included. It's really an honor to be here. So let me start. I'm focusing on diabetes, and you've heard a lot in the press about how diabetes might be a target disease for stem cell therapy. I'll focus on three things. One is the rationale for a stem cell-based therapy for diabetes. The second is where we stand now in generating insulin-producing cells for treatment of diabetics. And the third is a little bit about the ethical aspects of therapeutic cloning and the new options of iPS cells, as Clive briefly mentioned. It was a little bit unpredictable for me to get into the treatment of diabetes with stem cell therapy and stem cell research in general. And growing up a Catholic, I wasn't sure what I was, it was unpredictable that I was going to be going into stem cell therapy treatments. And this is something that was that's not going to work, excuse me. This was something that I hadn't planned on, so however, being a physician, I think that I come squarely down on the aspect of the ethical issues here between the moral status of the embryo and the moral status to relieve human suffering. I feel fairly strongly that there is really an imperative, and like Johnny here in the wheelchair, we need to consider that this is one of the ultimate goals and important things that we as humans can do is to relieve the human suffering. So everyone knows, I think, the public health impact of diabetes. It is a highly mortal disease. Patients, some die suddenly, but many die piecemeal from numerous complications. It is the leading cause of blindness and kidney failure. It accelerates cardiovascular disease, and patients ultimately have many comorbid conditions which lead to foreshortened lifespan. It's a very costly disease as well, and because of this, insulin had a major benefit in the 1920s when it was clinically applied for the first time. Insulin therapy relieved the instant death in patients with diabetes due to hyperglycemia and coma. However, it only prolonged the death of patients and is not really a cure. Diabetes comes in multiple types. For the purposes of this conference, we'll talk primarily about type 1 diabetes, which is due to the autoimmune attack of the insulin-producing cells in the pancreas and a very specific focused attack, which is focused on the beta cell or insulin-producing cell, which makes up about 1% of the pancreas, and that when that cell dies, it fails to produce insulin and our blood sugars are uncontrolled. It could also be applied to type 2 diabetes, where initially there's an insulin resistance and too much insulin produced to try to keep up, but ultimately there is a loss of insulin-producing cells and a relative deficiency of insulin. And in that case, it could also be applied. But probably for clinical purposes or clinical trials, the stereotypical disease is type 1 diabetes. So what are the current therapies? Well, there's insulin therapy, and a more state-of-the-art approach is this pump that you see here. And there's a continuous implantable delivery of insulin through this pump. And there's a sensor on the left here that talks to the pump and can deliver insulin and change the dose. There are some complications with current therapies, and in particular in this type of therapy there is not minute-to-minute regulation of the blood sugar, and in many cases patients still develop problems with vascular disease, blindness, and poor glycemic control leading to kidney problems and other end organ diseases. Pancreas transplantation, which has been around since 1966, is another clinically applied therapy that is something that is practiced at the University of Wisconsin and in selected transplant centers across the country. It is a better control of blood sugar. You can immediately normalize the blood sugars in patients within hours of surgery, in fact. You restore blood flow to the pancreas, and it turns on almost immediately, and insulin output starts. And by the time the patients are back to the floor, they have normal blood sugar control and don't need any insulin thereafter. Unfortunately, it's associated with some surgical complications, as well as some complications of immunosuppression, which is necessary to prevent rejection. And because of those issues, both technical and immunological issues, the gland or the graft does not last forever, and some patients have to return to insulin. In fact, the graft survival has improved, and it's a very successful therapy in some patients, yet other patients develop complications, including long-term complications such as infection and malignancy. So what are the other types of therapies that might be applied? Well, if one thinks about surgical therapies or transplant therapies, as in all cases of modern surgery, things are moving towards more minimally invasive approaches. And a minimally invasive approach for the treatment of diabetes is human islet transplantation. And you can see that here in the diagram of how this is done. You have the donor pancreas, and from that, one isolates by enzymatic and mechanical digestion the islets, again, which make up about 1% to 2% of the pancreas, and then purify them, separate them from the other parts of the pancreas, the acinar or enzymatic tissue, and then put them in a bag like grains of sand in an IV bag, and they settle by gravity, and by gravity they can be infused and take up residence in nearly any organ you choose. And it has been done experimentally in many different organs. In the humans, it's primarily done into the liver, because that is an available minimally invasive approach. So this is successful therapy in a number of patients. You can see here on the right some diagrams of glycosylated hemoglobin, which is a long-term measure of glycemic control. You can see this is in the Edmonton group data that shows that the blood sugars improve over time in these patients. And in the bottom panel on the right, you can see a day-to-day blood sugar measurements in one of our patients after an islet transplant, and you can see how stable they can be. So this is a very effective therapy in some patients. However, there are limitations. The limitations are due to a number of different things. For one, there's a shortage of donors. I think everyone is aware of that. And just recently, I think Aspirus got noted in the press for such a high organ donation rate, and one of the best in Wisconsin, so congratulations. In addition, it's quite damaging to remove islets from the pancreas, both from mechanical and enzymatic digestion. One can be overdigested, underdigested, and the mechanical trauma of the process can lead to long-term damage, and in fact, such damage that the quality of the islets may be suboptimal, and when one is implanting them, you get a suboptimal benefit. In addition, islets are highly immunogenic, and this, in fact, is perhaps even more immunogenic than a pancreas transplantation, and is potentially a major problem. It seems that islets might be more prone to the autoimmunity than a pancreas transplant. So we have islet transplantation as, at least in some patients, a successful proof of concept that cell therapy or a minimally invasive therapy might be beneficial or help a number of patients with diabetes and the lack of insulin. So you've heard quite a bit in the press again about how diabetes is a stereotypical disease that might be a target disease for cell therapies. But the question is why? Why precisely is that? Well, it's due because, for a number of reasons, it's due because there's a specific loss or a loss of a specific cell type. It's not multiple cell types that need to be replaced. It's one specific, one single cell type, the beta cell. In addition, unlike neural treatments or brain treatments where there's complexity and, as Clive indicated, a need to have careful integration of the cell you transplant. In the case of diabetes, that insulin production can occur anywhere in the body, and as long as it gets in the bloodstream, it'll work. And in fact, with a pancreas and islet transplant, the insulin is delivered in an atypical place or an ectopic place, meaning it's not delivered in the exact place where the native pancreas delivers insulin. So in that case, both with humans and experimental animals, we know that the site does not really matter. And the cell can be viewed as a simple protein factory that makes insulin. So with that, it can be produced anywhere, and there's less need for integration. In addition, as I mentioned, islet transplantation as currently practiced offers proof of principle, and there's a limited source of cells and tissues to treat patients. So here we have a proof of principle, and that's why diabetes is a target disease. We're working on ES cells as a cell source for diabetes treatments. Right now, many others are looking at other sources, including xenografts, but there are immunological hurdles that still are yet to be surmounted there. Potential for bone marrow as a source or umbilical cord cells as a source, many other investigators are looking at. That is still in its infancy and is a major question, although some success has been achieved experimentally. That has been called into question, and there's a lot of controversy in that field. From the point of view of the adult pancreas, one might ask the question, are there progenitor cells in the adult pancreas which then could be used to regenerate the organ, or beta cells? Unfortunately, they haven't been found yet, though many are looking, and it remains to be seen whether there are adult pancreatic progenitors that are capable of regenerating the pancreas. We do know that if a patient has a partial pancreatectomy or surgical removal of part of their pancreas and they become a diabetic, they never revert back to normal after that. So it's unlikely that there's robust regeneration. So when we entered this field a number of years ago, asking the question, can embryonic stem cells be used to produce beta cells, it was basically an idea of recapitulating development in a petri dish. And this is a diagram of showing how ES cells are derived from the blastocyst and then might re-enter a developmental pathway. Before I tell you a little bit about our work and where the field stands now, in the second part of the talk, I just want to briefly cover one important protein that is a master regulator of pancreatic development. And that is this protein shown here, pancreatic duodenal homeobox, or PDX1. It's the cells shown in green here as in the pancreatic bud, or the part of the gut tissue that branches to become the pancreas. And these cells in green are the forerunners of the pancreas, if you will. Without function of this gene, PDX1, patients and mice do not develop a pancreas and are born diabetic. So this is a key gene and a key protein to look for when thinking about can ES cells become pancreatic-like phenotypes. PDX1 is not only important in development at those earliest stages of progenitor cells, but it's also important in the beta cells and how the beta cell makes insulin. PDX is expressed in the beta cell as well as in the earlier precursor cells, and without PDX in the beta cell, those beta cells do not make a normal amount of insulin. And so it's needed at both the mature stage and the earlier stage. And at the mature stage, it's a marker of a true bona fide beta cell. So when we thought about asking the question a number of years ago, can ES cells become pancreatic cell types and beta cells, we took cells, ES cells, differentiated them in culture in the presence of serum spontaneously without any particular growth factors, and then stain the cells for these markers, insulin, other hormones that the pancreas produces, and this marker PDX1. And what we found was really, I think, surprising, but not surprising because we know that ES cells can become any cell type in the body. So this is mouse data. I'll show you human data in a minute, and basically what you see here on the left-hand panel is a cluster of PDX-expressing cells, those precursor cells, and then among those cells, some hormone or more mature cells are forming. And on the right-hand two panels, it shows that indeed some of those PDX-expressing cells become insulin-expressing cells, and they also express a marker C-peptide, which is another marker of bona fide insulin production or endogenous insulin production. So in fact, mouse ES cells can become spontaneously, at least some of the cells can become mouse insulin-producing cells. Human cells can do the same thing. Human ES cells can become PDX-expressing progenitors that also go on to express insulin and other hormones of the islet, namely somatostatin and glucagon. And that's shown here in the red cells expressing PDX and in green expressing insulin or C-peptide. However, we noticed that in looking carefully at these cultures, we saw many more progenitor cells that express PDX and very few insulin-expressing cells. In fact, the insulin-expressing cells were rare. And the left-hand panel shows a large population of PDX-expressing cells in red, but only 2 cells with the arrow that express insulin. And in fact, when we quantitated the numbers in the right-hand panel, we saw very few insulin-expressing cells and a little bit more of the progenitor cell markers. So why not very many endocrine cells? Well, if you think about it, the pancreas is not a large organ. It's only about 100 grams of a 70-kilo individual, and the beta cells only make up about 1% of the pancreas. And in fact, they're not as widely dispersed throughout the body as neural cells or skeletal muscle cells. And in fact, they're not necessary for normal in utero development. So what controls blood sugars in utero in a fetus, developing fetus? Well, it's the mother's pancreas, in fact. And in fact, PDX deficient mice, mice that don't have beta cells or pancreas, go through normal in utero development. So we know that these cells are not necessary for development or only necessary postnatally or after birth. So maybe that's why, and because they're so rare in the body, maybe that's why we're not seeing very many. So to summarize the data, and what I've showed you so far is that the mouse and human ES cells can form beta cells and pancreatic progenitors. Some of the aspects of the characteristics of the cells seem to be a normal, mature characteristic beta cell. We know that also found that high serum conditions seem to inhibit this differentiation process in culture. But we also know that so far we don't have enough beta cells to really transplant them or to expect they'll have a benefit or produce enough insulin to reverse diabetes in an animal. Now, not only we, but others have found similar findings in human and mouse ES cells, but where the field stands now is really to get a robust population, a larger population of cells, and this is the current challenge. So we have, and this diagram here represents what we think is going on, and that is we have ES cells, and they start to differentiate under some conditions if they're not targeted conditions or guided or directed differentiation conditions with specific growth factors, that under non-selective conditions they might form multiple different cell types, some of which may be pancreas committed, but others are committed to neuroectoderm and mesoderm and other cell types. So we're interested in getting rid of those for the purpose of treating patients with diabetes, and how do we do that? Well, one is to take signals and clues from embryonic development of what molecules and what genes control this process during development, applying that to the cells in culture. So many people are looking at this. However, I think a rational approach, or this clues from development approach, is not going to be the entire answer, because if we look for mouse and zebrafish development, that might not be the same as what happens with the molecules and genes that control things in humans. So we need to have a broader approach and apply some degree of a shotgun approach or empiric approach to this as well. So we and others are looking at this, and I'll just give you one example of each, empiric approach and a rational approach. First, the rational approach. Some investigators in Sweden took the idea that if there's some developmental signal in the embryonic pancreas controlling differentiation, let's mix ES cells, human embryonic stem cells, with a mouse developing pancreas at an early stage and see if those signals will guide the ES cells to differentiate to the right direction. Well, they in fact found that. So the experiment they did is as shown here, there's no needle in the eye here, Clive, so no one has to wince here. So they took human embryonic stem cells that were partially differentiated towards endoderm, studied by a number of markers, and hand-selected out those cells in a non-purified way, and then co-transplanted them with an embryonic mouse pancreas in an animal and let them differentiate there for a few months. And after some time, they took out the tissues and stained the cells. Prior to transplanting the cells had endoderm markers or precursor markers such as PDX, but they did not express insulin in their hands. But after transplantation, they saw some human cells, not mouse cells, expressing insulin, because there are going to be some there, but human cells expressing insulin by the markers of green for insulin and human nuclear marker in red there. So you can see in that panel that there are some human cells that have presumably human embryonic stem cells that are now expressing insulin, glucagon, and amylase, an enzyme that the pancreas produces. So here we have evidence that there may be signals from the pancreas that are specific in guiding differentiation of the endoderm towards an endocrine cell type that produces insulin. I should mention that when they did the same thing in mixing the cells with brain tissue, they did not get the same result, showing the specificity. So another group has taken a partially empiric approach and a partially rational approach by taking some growth factors and mixing them with human ES cells in a sequential fashion, some growth factors that have specific activities, one called activin, for instance, that is a nodal stimulatory molecule, and this stimulates primitive streak development, which is the beginning of when the embryo decides to become different cell types. So using activin, applying it to human ES cells, and stimulating nodal signaling, they were able to achieve in a stepwise fashion some enrichment of endoderm, some enrichment of pancreatic precursor cells expressing PDX, and a population of insulin-expressing cells. And in fact, their insulin-expressing cell population is the greatest number in terms of percentage of cells achieved to date, and that's shown here in the left-hand two panels. And you can see with low activin treatment, only a few cells, 0.3%, were stained with insulin in green. And with a larger dose of activin, a significantly greater percentage of cells were expressing insulin, in fact, 7% of the population. Now that was a notable achievement. However, it still doesn't represent an enriched population. And still the field is in a state where we're not quite there yet. In fact, the cells, although they're 7% or 10% of cells that express insulin, they have some characteristics that are not particularly mature. And these are some of the scientific characteristics. I won't go through all the details there, but in fact, they have low stimulated glucose, or low glucose stimulated insulin secretion, which of course is a hallmark and an absolute necessity for a beta cell. So in fact, it's suspected that these may represent immature beta cells. So the field is at a stage where we can see that there is able to have beta cell differentiation from embryonic stem cells. The question is, how do we get an enriched population and how to get them better functioning? There are a number of other challenges and hurdles left to be obtained or achieved before we go to clinical therapy. Whereas clinical therapies are nearing initiation for spinal cord diseases, as Clive mentioned, for diabetes, I think they're farther away. And these are some of an outline of some of the generic problems or issues that still need to be faced and addressed. Not least among them is the safety issues of forming teratomas and tumor formation and adverse effects that might occur of unwanted tissues being transplanted. But also we need to show efficacy in an animal model of diabetes. And that's where the field stands now. So that gives you a summary of where the diabetes field stands. So in the last 10 minutes or so of the presentation, I just wanted to talk briefly about the aspects and ethical aspects of generating patient-specific embryonic stem cells. So it's been talked about in the literature that getting patient-specific embryonic stem cells is the home run. And in fact, since the early days after Jamie Thompson's discovery in 1998, and Dolly's cloning and shortly thereafter, it was realized that this might be a very elegant approach to obtaining genetically matched or histocompatible embryonic stem cells for transplantation. And the advantages there are that it can obviate the need for immunosuppression and eliminate the possibility of rejection. So those are the advantages, and whether they are really capable of avoiding rejection, avoiding the need for immunosuppression remains to be seen, but there are a few animal studies that suggest that they would not succumb to rejection processes. So the approach, or one strategy, of course, is somatic cell nuclear transfer and nuclear cloning. I won't talk about these other potential strategies and just focus on somatic cell nuclear transfer or therapeutic cloning. As it's been envisioned and is shown in this diagram here, and what one can see is starting with the potential patient on the bottom, one would remove a somatic cell, take the nuclear material from that, put it into a third party, a nucleated oocyte, basically cloning to create Dolly, and then go through some development in vitro to develop a blastocyst. From that cloned blastocyst, one could generate a embryonic stem cell line, a new embryonic stem cell line, and then differentiate those tissues in culture to the cell type of interest and then transplant back those to the patient in need. So that, in essence, is the cycle of therapeutic cloning. And previously in the press, there was a lot of discussion or confusion about reproductive cloning versus therapeutic cloning. I think we all agree there are potentially disastrous consequences of reproductive cloning. However, I wanted to impress upon you, this audience, both ethicists and scientists alike, that there might be some very beneficial consequences of therapeutic cloning. And the proof of principle of how this might be used in the clinic is shown here in this very elegant animal experiment. And I'd just like to go through this cycle briefly with you. So this group in Boston, what they did is set out to correct the immunological defects of an immunodeficient mouse, the RAG2 knockout mouse, which fails to develop T and B cells and is basically like the boy in the bubble, or immunologically depleted. So they set about making new hematopoietic cells or blood cells and then put them back into the mice to cure it. So what they did is took a series of cells from the tail of that animal, fibroblasts, and then performed nuclear transfer in a way I just showed, and then to generate a cloned blastocyst, and from that blastocyst that would be genetically identical to that RAG2 deficient mice, generated embryonic stem cells. And those defective embryonic stem cells now, they set about repairing the genetic defect in those embryonic stem cells, and they did that by common genetic approaches, not requiring viruses in this case, but homologous recombination. And they repaired the RAG2 defect and replaced it with a normal RAG2 gene. And then they differentiated the cells and put another transcription factor in the cells to stimulate blood development, and after that generated blood cells and hematopoietic stem cells that they were able to transplant back in the animals to correct the defect, and they showed a fairly robust correction of the immunological defect. So this shows that a combination of somatic cell nuclear transfer and cloning and gene therapy could correct a genetic disease and not only supply cells to replace deficient cells in certain diseases that we've been talking about, but also repair a genetic disease. So I think this is a very elegant approach, could have wide-ranging implications for human therapies. So what are the problems, though, with somatic cell nuclear transfer or therapeutic cloning? And they really arise from the two major issues here, and that is the source of oocytes or eggs that are necessary to make a cloned blastocyst, and the second is the generation of an embryo that's earmarked for destruction. So because of those two ethical problems with somatic cell nuclear transfer, it boils down to this has generated a whole cottage industry in science in and of itself to deal with this political and ethical issue. And there's been some fairly innovative approaches taken. I've outlined a couple here. None of these, the first three, that is altered nuclear transfer, which generates a blastocyst that is not able to be implanted and therefore one could consider it abnormal, or blastomere-derived human ES cells, or parthenogenic-derived ES cells. None of these actually fully deal with the ethical issues of the source of oocytes and the generation of an embryo. With somatic cell nuclear transfer, we need a large source of oocytes because the current inefficiency of the process. And where does one get those? One gets those from normal females that are hyperovulated. They have to go through a potentially risky and uncomfortable procedure. And this is ethically difficult if there is no benefit to that individual. That's what was done in Korea, and there has been political fallout, as you know, about that. And to date, although I know a number of people, investigators in the US and worldwide, are working on improving techniques of somatic cell nuclear transfer, of course, there's been the political issues with that in the Korean group a few years ago. Parthenogenetic ES cells might generate a genetically identical ES cell, but then it would be just a match to the female that the oocyte was derived from. And this doesn't serve the other half of the population. And blastomere-derived human ES cells would only serve the individual who was the other part of the embryo. So it's for a future individual patient that would be born by that IVF approach. Really the landmark achievement in this field to take the field a step further has been with induced pluripotent stem cells. As Clive mentioned, both the Madison group and a Japanese group achieved this this past year in human induced pluripotent stem cells after a few years ago achieving this in mice. And the way this works is diagrammatically here, a somatic cell from the individual prospective recipient of a therapy. In this case, Dr. Thompson and Dr. Yu used fibroblast cell line and fetal foreskin fibroblasts to do this. They did not use other cell types, other somatic cell types, and we'll get to that in a minute. And what they did is overexpressed 4 genes in those cells. These are reprogramming genes or pluripotency genes, OCT4, SOX2, NANOG, and LIN28. And after overexpressing these transiently in these cells, the cells seem to take on characteristics of pluripotent cells and were able to proliferate and have all the markers of standard ES cells. In fact, they were so similar when they compared them by DNA microarrays, comparing hundreds of thousands of genes, or tens of thousands of genes, they were virtually identical to human ES cells. So what they did was, as shown here on the right, take the fibroblasts in panel B on the right-hand side there and turn them into iPS cells by overexpressing the genes. You can, I won't go into the details there, but 3 genes were critical which overlapped with the Japanese group. But will the iPS cells be useful clinically? I think that's a major question and remains to be answered. I think there are some issues and some scientific questions still need to be addressed. Those are, as outlined here, first related to the source of the fibroblasts. The source in their experiments were neonatal foreskin fibroblasts and a fibroblast cell line. And of course, many of the patients needing this therapy are adults, and it remains to be seen whether their fibroblasts would have the same ability to be reprogrammed as a fetal or young fibroblast. It is also a question whether other somatic cell types, skeletal muscle or other tissues, might have that same ability to be reprogrammed by the same set of genes. So I think that needs to be addressed to think about applying this widely and clinically. The other is that these are genetically manipulated cells. These are not natural cells. They have lentivirus, which is an HIV-type virus, used to transfect the cells and express these genes. And this is potentially problematic from an issue of safety of gene therapy. So, and it also, the genes that were put into the cells might interrupt the functions of normal developmental genes, and therefore it might interrupt normal development. And then the question arises, can these iPS cells differentiate normally in the normal pathways that human ES cells do in culture? And would they be responsive to the same signals and become the same cell types that we're interested in transplanting? So these are some of the critical questions, not the least of which is, will iPS cells be a model of human development like human ES cells? Since they're highly manipulated and they're not embryonic in their start, will they be a model of human development? I think we'll need to do studies to compare human ES cells and iPS cells in the future to really sort that out. So I think we've covered a few things. One is the rationale for using ES cells as a therapy for diabetes. Two is where the field stands now in terms of generating functional insulin-producing cells for therapies, how that might be applied clinically, what the hurdles are and what the current challenges are, and a little bit about the aspects of iPS cells and creating histocompatible cells for transplantation, and where the field needs to move in the future with regard to iPS cell studies. This last slide acknowledges some of the people in my lab who did some of the work and our funding sponsors, and we're very grateful to our funding sponsors and for all the scientists in the lab. Thank you very much.